Okay, so now we're recording, and I'll share the PowerPoint again. So this unit covers mainly metabolism, but it starts with an overview of nutrition, which is important to have a good sense of after we've now talked about the digestive system and before we get into metabolism. So the nutrition section will go through just the basic nutrients that are ingested and what their uses are in the body once they get broken down by the digestive tract. And then basically what metabolism covers is after digestion occurs, then what happens. So we talked about with digestion, the structures that are involved in ingestion, in moving food through the GI tract, in breaking down food, also the enzymes and the chemicals that are involved in that, and then absorption a little bit, and then we kind of stopped there. So this really starts off where the digestive system left off. After nutrients are absorbed, then how are they used in the body? So that's what we'll start to get into today. And rather than trying to hurry up and get through the entire outline, I think what I'm going to do is we will take our time, get through what we can today. And then I am working next week during spring break. So I will record a short lecture for some of the key things that we don't finish today. So don't worry, we probably are not going to get through the entire outline today, but I will do a recorded lecture. And I'll definitely make it available earlier in the week. So you guys will have it for at least a week before you have your test on this stuff. All right, so let me get started. And I'm, again, going to do what we typically do with the lectures. I'm going to go through the lecture outlines, and we're starting with the first one from Unit 4. So it's the nutrition and carbohydrate metabolism study outline. And that's the one that begins with an overview of nutrition and nutrients. So it does explain what a macronutrient is. These are things that you need a lot of in your diet versus micronutrients like vitamins and minerals. But now I will start by going over the 5 main macronutrients, starting with carbohydrates. And most of these are derived from plants. There are the simpler, smaller sugars, which are monosaccharides and disaccharides. These typically come from fruits, from milk, like lactose is found in milk, and also honey. And there are also polysaccharides, which are the much bigger complex carbohydrates. These come from grains and vegetables, typically in the form of starch or fiber. And then speaking of fiber, in your outline, you have definitions for the 2 main types of fiber, soluble and insoluble fiber. And just explains the difference and where they're found. Soluble fiber you can get from several different sources, like apples, pectin found in apples and oranges, for example. And that can be digested, but insoluble fiber can't be. And that's found in cellulose, which is in a lot of vegetables. As far as uses in the body, carbohydrates are primarily used by cells to produce ATP. So ATP stands for adenosine triphosphate. You would have for sure talked about this in A&P1. We are going to talk about it a lot more now that we're getting into metabolism. But just for now, know that ATP stands for adenosine triphosphate. That is really the only form of energy that your cells can directly use. So generally, all carbohydrates are converted into ATP for your cells to use for energy. And then as far as the dietary requirement, this is just the percentage that these should make up your diet. And they should be anywhere between like 45 and 65% of your total calorie intake. So on average, it should make up a little bit over half of total calories ingested. Okay. So next ones are the lipids. These are fats. And as far as dietary sources, triglycerides are the most abundant type of lipid found in our diet. There are saturated and unsaturated fats. Saturated fats come from typically meat, dairy products, and hydrogenated oils, which, like, for example, would be something like shortening, like Crisco or margarine. And then there are also unsaturated fats. These are like the more healthy fats. These come from seeds, nuts, also olive oil and other vegetable oils. Then there is cholesterol, which it comes from egg yolks, also meat, shellfish, and dairy products. Okay, and then finally, there are essential fatty acids, which are really important to have in your diet. And you've probably heard of these. They include the omega-6 and omega-3 fatty acids. So as far as uses in the body, lipids, unlike carbohydrates, which primarily have just one use in the body, and that's for energy, lipids have a lot of different uses in the body. First of all, they can serve as a source of energy, but typically only after glucose, which is the primary form that carbohydrates would be in when you're using it for energy. So typically, fats are only used for energy after you've used up all the glucose that you have available for energy. Also, fat found in adipose tissue provides insulation and cushioning around the internal organs. So it provides like a protective layer around major internal organs. Another type of, a specific type of lipid are the phospholipids. They form the myelin. If you guys remember learning about neurons, remember there is a myelin covering around parts of a lot of neurons. So they form myelin, and also phospholipids form the plasma membrane that surrounds, it forms the outside of all your cells. Okay, there's a couple more functions as well. Absorption helps with the absorption of fat-soluble vitamins. There are certain vitamins, they're called fat-soluble vitamins, that have to be absorbed along with fat. Otherwise, they can't be absorbed. So fat helps, therefore, with the absorption of these types of vitamins. And then finally, cholesterol is used to make bile salts. Remember, that's the main substance found in bile. And also, cholesterol helps to form steroid hormones. That's a particular type of hormone, like a particular class of hormones. So cholesterol forms the basis of those. Okay, and then finally, the dietary requirement for lipids is that they should make up anywhere between 20 and 35% of total calorie intake. Okay, and now proteins. Let me go to the next slide. So proteins can come from animal products and non-animal products. Animal products typically provide the highest quality protein. And when I say a high quality protein, it means that the protein contains a lot of different amino acids. There's 20 different amino acids that exist. So a high quality protein would contain almost all 20 or all 20 different amino acids. Non-animal products include things like legumes, which are beans, nuts, and cereals. They provide a lower quality protein. So that just means they don't contain quite as many different amino acids. For uses in the body, proteins also perform a lot of different things. They provide structural materials. So think about things like collagen. Collagen is found in a lot of different connective tissues. It's in hair, it's in skin. Keratin is another one that's found in skin, hair, nails. And another example is like the proteins found in your muscles, actin, myosin. Another use in the body is that they can act as enzymes and hormones. And then finally, they can serve as a source of energy, but typically only if there's not carbohydrates or fats available. So your body is always going to use carbohydrates first for energy, then fats. Then if it has to, it's going to use proteins, but that's kind of like a last resort thing. So for the dietary requirement, 10 to 35% of total calorie intake should be protein. And related to protein is a term called nitrogen balance. So proteins, this is different from carbohydrates and lipids. They contain nitrogen. The other two main organic molecules, the nutrients that we ingest, do not contain nitrogen. So proteins are a main source of nitrogen. So that's why this is called nitrogen balance. And basically the rate of protein synthesis, like the rate that you're making new proteins, should equal the rate that proteins are being broken down. And that would give you equilibrium. That would give you like an equal nitrogen balance. If there is a positive nitrogen balance, that means that protein synthesis is greater than decomposition. So you're making proteins at a faster rate than you're breaking them down. And that's often seen during times of growth. Like if a child has a growth spurt, for example, or during times of like tissue repair. So you're growing a lot of new tissue. On the other hand, negative nitrogen balance is when protein synthesis is less than the rate of decomposition. So your body is breaking down proteins more than it's making them. And that's not a good thing. This is often seen when individuals are suffering from a serious injury or malnutrition or starvation, or even like a really high fever. Remember, high temperatures can break down proteins, that denatures them. So that is a bad thing. Okay, so now for the micronutrients. Micronutrients are things like vitamins and minerals. Vitamins are organic compounds, and they're not used for energy, but they can help out the body perform different reactions, and they can work with enzymes to help with reactions. So vitamins are classified based on their solubility. There are, oops, let me change the slide, sorry. There are water-soluble vitamins and there are fat-soluble vitamins. The water-soluble vitamins you can see on the PowerPoint are the vitamin C, and then the B vitamins. So the fact that they're water soluble means that they are absorbed along with water in the digestive tract. And also, any amount that is not used pretty much right away will be excreted in urine. So excess amounts of these vitamins are not stored. Any amount that's not used, like within an hour maybe, would be excreted in urine. Okay, on the other hand, there are also lipid soluble vitamins. And you can see on here on the outline or on the PowerPoint that these include vitamins A, D, E, and K. So these bind to ingested lipids and they are absorbed along with those lipids. So when you eat fats, eat lipids, and they get absorbed, fat-soluble vitamins get absorbed along with them. And the other difference here is that these can be stored. They get stored in the liver and in fat, in adipose tissue. Okay. So there's two terms below here, hypovitaminosis and hypervitaminosis. Hypo, that means the prefix hypo means like less than, under, below. So this is when you don't have enough of a particular vitamin. So vitamin deficiency. And this can happen with a lot of the different vitamins. I've just given you a couple of examples that result in certain conditions or diseases. There's way more examples than this, but these are two commonly seen examples, especially in countries where people don't have access to good, healthy foods in a lot of times. So one example is when there's a deficiency of vitamin D that can lead to rickets. And on the PowerPoint, you can see the result here on these pictures. With rickets, you have decreased calcium absorption, which leads to the softening of bones, and that causes bone deformities. Okay, so that is what you're seeing here on this picture. This is mainly showing you like the femurs of an individual, and normally they're perfectly straight. But in somebody with rickets, because the bones are soft, when you're consistently putting weight on the femurs, which are the major leg bones, the bones in the thigh, they tend to bow out because these bones are soft. They don't have enough calcium in them to have calcification. So in that way, you have this bone deformity. And another example is if you have a deficiency of vitamin B12, that leads to pernicious anemia. And this is usually caused by the inability to absorb vitamin B12. It's not that it's lacking in the diet, but the individual is unable to absorb it properly in the digestive tract. So the result of that is that they have fatigue, weight loss, often difficulty concentrating. And on the other hand, I didn't mean to do that. Let me go back. I don't have a separate slide for hypervitaminosis, but this is the opposite situation. Rather than having a vitamin deficiency, you have excessive amounts of a particular vitamin. And that can be harmful if the intake of a particular vitamin exceeds the body's ability to use and store that vitamin. So if you're intaking like a really high amount of a particular vitamin and it's being stored in the body, it can actually build up to toxic levels. So too much of a particular vitamin isn't necessarily a good thing. A lot of times people think they take all these vitamin supplements and think that it can't cause any harm, but it actually can, depending on the particular type of vitamin and how much it is being stored in the body. Okay, so the other micronutrient are minerals. These are a little different from vitamins because vitamins are organic compounds, but these are inorganic. That means that they don't contain carbon and they're typically smaller than vitamins. You have in your outline a list of the ones that have really a vital role in the body, things like calcium, sodium, potassium. They have roles in sending nerve impulses, muscle contraction. So they are really important, obviously. And like I just said, they have vital roles in the body, but they're not used for energy. They're used for other things, for performing certain functions. Okay, so we are pretty much done discussing major nutrients and nutrition. Now we will get into metabolism, starting with just like an overview of what metabolism is and particular types of reactions. So once nutrients that we just talked about, carbohydrates, lipids, proteins, are absorbed by cells, nutrients they're involved in a lot of different types of reactions. And together, all the reactions that are performed on these nutrients is called metabolism. And there are two just really big, like general reaction types that are involved. You guys have the definitions for these already in your outline: catabolism and anabolism. Catabolism is, you can see an example of this on the right side of the PowerPoint slide. We're taking a large molecule and you're breaking it apart into smaller molecules. And a lot of times that will release energy when you do that. The opposite of that is anabolism, or anabolic reactions, where you're taking smaller things, smaller cells, smaller molecules, and you're putting them together to create one large molecule. And a lot of times, if you're putting those things together, you're creating new bonds. That requires energy to do that. Another set of reactions next in your outline, which you don't have a definition for, so I'm going to give them to you, is the oxidation and reduction reactions. So oxidation involves the removal of electrons and hydrogen ions from an atom or a molecule. One additional thing to note is that when those things are removed, it also removes energy. It's taking energy away from the atom or molecule. When you remove electrons and hydrogen ions, you're also taking away energy. So it's an energy loss reaction. So if you can remember oxidation, reduction is the exact opposite. This is the gain of electrons and hydrogen ions by an atom or molecule. Okay, and that is an energy gain reaction. When you're giving an atom or a molecule electrons and hydrogen ions, you're also giving it energy. And most times, these reactions happen simultaneously to two different molecules. So you will have one molecule that's getting oxidized, you're taking away electrons and hydrogen ions. And at the same time, you have another molecule that you're giving those things to. So that molecule gets reduced. So when you're oxidizing one molecule, you're reducing the other one. You're taking electrons and hydrogen ions away from one and giving them to another. So almost always you're going to see that happening, that those two reactions are happening at the same time to two different molecules. One is losing electrons and hydrogen ions, and the other one's gaining them. Okay, going down in the outline, coenzymes. These often work with enzymes during the redox reactions, the reduction and oxidation reactions. You guys have information in there already in your outline. Let's talk about the important coenzymes. There's 3 of them. You have almost everything filled out for, I think, NAD and FAD. I am always going to use those terms for them. Whoops, let me go back. Because their real names are really long. I wrote them out in your outline, but I'm always going to just refer to them as NAD and FAD. So NAD is the oxidized form, the form that does not have the electrons and the hydrogen ions. It can be reduced, so you can give it electrons and hydrogen ions, and then it becomes NADH plus an additional H. So if you see H's on NAD, you know that that is the reduced form because it's been given hydrogens. And remember, when you are reducing a molecule, when you're giving it electrons and hydrogen ions, you're also giving it energy. So now when you have that form of NAD, when it's NADH plus H, and it's reduced, that has a lot of energy. It's a high energy molecule, and it contains the potential energy to eventually form 2.5 ATP molecules. FAD is the other one, and it's very similar. So FAD is the oxidized form where it doesn't have electrons and hydrogen ions, but it can be reduced. So you can give it electrons and hydrogen ions. And when you do that, it's called FADH2. So it has the H's on there. So you know that that's the reduced form of it. And FADH contains the potential energy to eventually form 1.5 ATP molecules. Okay, so basically these 2 coenzymes, they can be reduced, which means that you've given them hydrogen ions, electrons, and energy. And that energy can be used to make ATP. I remember we talked about a few minutes ago the fact that ATP, it's a very valuable molecule because it's the only molecule that can be directly used by your cells for energy. Okay, so ATP is very important. It is the main source, really, actually, the only source of energy in the body. Okay. And then the third coenzyme is called coenzyme A. It is not involved in these redox reactions. It doesn't get oxidized. It doesn't get reduced. It just serves as a carrier molecule. So that means it carries another molecule, like from one place to another. It's like a transportation system. It's like an Uber for other molecules. Okay. Now, this next little section is just giving you guys some definitions for other reactions that you'll see that are involved with metabolism. And a lot of times the name indicates what is going on in the reaction. The first one doesn't really, but the next couple kind of do tell you what's happening. The term isomerization means a change in the structure of a molecule without a change in its chemical formula. So the chemical formula tells you what atoms or how many atoms of each element you have in a molecule. So the chemical formula, for example, if you're looking at these 2 pictures for glucose and fructose, is C6H12O6. That tells you that you have 6 C's, which are carbons, 12 H's, which are hydrogen atoms, and 6 O's, which are oxygen atoms. C6H12O6. That's the chemical formula. So with isomerization, you're not changing the chemical formula. That means you're not adding any more atoms. You're not taking away any atoms, but you're rearranging them. So you're like putting them in different order in the molecule. So like if you look at this picture, you can see for glucose, the oxygen is double bonded to the very first carbon in the molecule. But in fructose, the oxygen is double bonded to the second carbon. So you've just rearranged the atoms within the molecule a little bit. You haven't added anything or taken anything away. You're just rearranging it. So that's all that isomerization is. Hydration, if you guys look at this diagram, you can probably see what's happening with hydration. See what's being added? Water. That's the addition. It's the addition of a water molecule. Dehydration is just the opposite, removal of a water molecule. Okay, decarboxylation. If you look at the diagram, you can probably see what's happening here. It's removal of carbon dioxide, CO2. And then the last two, phosphorylation and dephosphorylation, these are opposites of one another. Phosphorylation is the addition of a phosphate group. And dephosphorylation is the removal of a phosphate group. All right, so I said we would talk a little bit more about ATP. You guys already know that it's the primary source of energy for the body. It's called, I think your textbook calls it energy currency. It serves as the energy currency for cells. So it's really like the only form of energy that cells can use. You can see its basic structure here. It has three main components. It has a sugar, so just a simple sugar right here. It has adenine. That's just a molecule that contains nitrogen. And then it has three phosphate groups attached to it. Okay, and now there's two main methods of producing ATP. Your body is always producing ATP because you constantly need it, because your cells constantly need energy to remain alive and functioning. So you are always making ATP. There is substrate level phosphorylation, and then there's oxidative phosphorylation. Those are the two main ways of making it. With substrate level phosphorylation, this is when ATP is made by transferring a phosphate group from a molecule to ADP. Okay. So basically you're taking a phosphate group from one like random molecule, and there's certain molecules that are typically used a lot to do this, but from one molecule, and then you're giving it to ADP. When you give a phosphate group to ADP, it becomes ATP. One of the most commonly used molecules to perform substrate level phosphorylation is creatine phosphate. And this is in your outline, that's written there. So creatine phosphate often donates a phosphate group. It gives it away and it gives it to ADP in order to make ATP. So that is what you're seeing here. You have creatine phosphate. It is giving a phosphate group to ADP to make ATP. So your body typically has a store of creatine phosphate, but you're unable to store a whole lot of it. So typically you only have enough creatine phosphate to make ATP that will last for about 15 seconds worth of activity. After that point, after you used up all that energy made from creatine phosphate, then you run out of it. And then you need another method of making ATP, because usually creatine phosphate, you don't have a lot of it, so it's used up pretty quickly. So the other method that can be used for making ATP, and this can be used for a much longer period of time, is oxidative phosphorylation. And this occurs during cellular respiration. This occurs during cellular respiration, when energy is captured during the breakdown of food to make ATP. So this graph just gives you an example of like the length of time that these two methods of making ATP can give you energy for. So the blue line, I'm really just going to focus on the blue line and the green line. The blue line shows you the number of seconds that this method of making ATP will give you energy for. So using creatine phosphate, you will get energy right away. But it only lasts, like you can see, not even a minute, not even for 60 seconds. And then you use up all your creatine phosphate and then you can't get any more energy from it. So at that point, your body turns to oxidative phosphorylation, and it's slowly using other molecules from the nutrients that you've ingested in order to make ATP. And that can last for a really long time. So that is now the main thing that we are going to focus on. We're going to focus on oxidative phosphorylation. So that is using the process is called cellular respiration to use the nutrients that you've eaten from the foods that you've eaten to make ATP. Okay. So maybe this is a good time to take a little break, and then we'll come back and I can talk about, I'll talk about cellular respiration. And I think we'll get through cellular respiration probably, and then I'll stop there. And for the rest, I can just do a short lecture recording for you guys to do the rest of the outline. So let's take a break. It's 10:12. Let's see, we can come back at 10:20 and go until like about 11. All right. So I will see you guys back in about 8 minutes. Okay, it's 10:20, so I'm gonna start back up where we left off. Sorry, let me pull up the outline. Okay, so we introduced cellular respiration, the main process for making ATP. It involves breaking down glucose, which is a simple sugar. Any carbohydrate can be converted into glucose. It can be easily converted in like one step into glucose. And even other types of nutrients like lipids can be converted into like a form of glucose. And that is what gets broken down to release energy that is then used to make a large amount of ATP. Couple things about the process: it requires oxygen, and it results in the production of either 30 or 32 molecules of ATP. It actually depends on the type of cell that is making the ATP. There are certain ones that can make a little bit more. So that's why there's 30 or 32 that can be produced from just a single glucose molecule. And then byproducts of this process are carbon dioxide and water. So this is there's four main steps within the overall process of cellular respiration. And it starts with glycolysis. This is step one. And this is the only step that does not require oxygen. So the term for that when a process does not require oxygen is anaerobic. So this process is anaerobic. It occurs in the cytosol of cells. The cytosol is just the liquid part of the cytoplasm. Remember, you have a cell and it's filled with cytoplasm. And then there's all these different organelles within the cytoplasm. So the cytosol is just the liquid part that surrounds all those organelles. And then the function of glycolysis, so what's actually happening during glycolysis, is that this step produces 2 ATP through substrate level phosphorylation. Also, small amounts of energy are collected from the glucose molecule and are stored for the last step of cellular respiration. So during the process of glycolysis, glucose is modified and broken apart. It's actually split into 2 pieces, and those 2 pieces are modified. And then ultimately, glucose becomes what's called pyruvic acid. So you're starting out with 1 molecule of glucose, and you're ending up with 2 molecules of pyruvic acid. The entire next page of your outline shows you the reactions that are involved in glycolysis to break down and modify the glucose molecule. I know it looks kind of crazy and intimidating, but I mean, the main things to know are that you're starting with a single glucose molecule. It's being broken apart, it's being modified, and what you're ending up with after all those reactions take place are 2 separate molecules of pyruvic acid. One important thing to make note of is initially to get this process going, you actually have to use up ATP. So if you look at the diagram on the slide, you need to use up 2 molecules of ATP. It's going into the reaction to get it going. So initially, you're actually negative 2 ATP molecules. But then as this process continues, you do end up making 4 ATP molecules. You make 2 here and 2 here. So that's 4 total. And then if you take into account that you used up 2 in the beginning, that tells you that your net gain of ATP after this process is 2. And we wrote that down when we discussed the function of glycolysis. I said that this step does produce 2 ATP molecules right away through substrate level phosphorylation. Okay, and that's because you use up 2 in the beginning and you make 4. So 4 minus 2 gives you 2. So right away, this step will give you 2 ATP molecules. It's not a lot, but it gives you a little bit immediately. The main thing, though, is that during this process, you are basically taking energy away from the glucose molecule, and you're giving it to one of the coenzymes that we talked about more towards the beginning of the lecture. Remember NAD and FAD? We talked about how they can be oxidized and reduced. Well, here's NAD. It's involved in this process. NAD comes in and it gets reduced. You are giving NAD electrons and hydrogen ions and energy from the glucose molecule. So you are taking NAD and you're reducing it by giving it electrons, hydrogen ions, and energy. So by doing that, you're basically taking energy away from the glucose and giving it to these coenzymes. And these coenzymes are going to do nothing with that energy right away. They are storing it. They just hold on to it. They just become high energy molecules. But during the very last step of cellular respiration, step number 4, all of these coenzymes that get reduced, that are being given energy just to store temporarily, and this is going to happen in steps 1, 2, and 3. In step 4, all of the coenzymes that are storing energy are now going to come in and give up that energy to make a whole bunch of ATP. So it's in step 4 that all the coenzymes come, give up their energy, and you get to make a whole lot of ATP there. So that's what happens during glycolysis. At the end of glycolysis, if there is sufficient oxygen, if there's oxygen present, then the process will continue. Pyruvic acid is going to enter the next step. It's going to go into step 2. Because oxygen is needed for all the other steps of cellular respiration. This is the first step, is the only step where it's actually not needed. But if there's insufficient oxygen levels, and that could be the case during really strenuous exercise when your muscles are using up almost all the oxygen that's available, in that case, pyruvic acid is converted into lactic acid. Okay, so pyruvic acid is converted into lactic acid, and you actually can't continue on with cellular respiration. Once oxygen levels are restored, it can be converted back to pyruvic acid, and then you can continue. But temporarily, the process is kind of halted until you get enough oxygen to be able to continue on to step 2. So in step 2, I'm going to come back to that picture that I had up, but step 2 is the oxidation of pyruvic acid. This step, as well, along with step 3 and step 4, these require oxygen. Step 1, glycolysis, is the only one that doesn't. And for the location of where this takes place, remember, glycolysis occurs just like in the middle of the cell, in the cytosol, but this occurs within the mitochondria. And let me go back now. This is a picture of a mitochondrion. Remember, there are various organelles within a cell. They each have a specific function. You learned about that in the beginning of A&P 1. So mitochondria are one type of organelle that's found within cells, and within the mitochondria is where a lot of ATP gets produced. So specifically where this process occurs, it's inside of this structure, but it's in the inner matrix, which is the fluid that's all the way in the middle here. So this process occurs within the inner matrix of mitochondria. So that's the fluid that's in the very center of that structure. Okay, and this step is just kind of like a transition step between glycolysis and step 3. Pyruvic acid, which is what came out of step 1, can't enter step 3. So it just has to be converted into something that can go into the next step. So this is really just like a quick conversion step where you're converting pyruvic acid into a different type of molecule. There's no ATP that gets made during this step, but we are collecting small amounts of energy from what's left of the glucose. Now it's called pyruvic acid. And that is stored just like in glycolysis. You're collecting energy and it's stored for use in the last step of cellular respiration. So in your outline, you do have that picture of a mitochondrion, so you can see the different parts of it. And then you have this exact representation of step 2, the oxidation of pyruvic acid. So this step is very simple. There's only 3 reactions that occur. With the others, there's a lot more. So in this one, you're taking, remember, after glycolysis, you ended up with 2 molecules of pyruvic acid. So this is showing you one of them, but keep in mind that this is going to happen to both of them. So each pyruvic acid molecule, which looks like this, is going to undergo these 3 reactions. It's going to undergo decarboxylation, which remember is removal of carbon dioxide. It's going to get oxidized, which means that you're taking away electrons and hydrogen ions and energy from it. And you're giving those things to NAD. Again, this is what happened in glycolysis. So you're reducing NAD to make NADH plus H. And this is now a high energy molecule. It's storing energy that's going to be used in the last step. And then finally, you just add coenzyme A. Remember, coenzyme A doesn't really do anything. It's just a carrier molecule. So it can carry this little acetyl group here into the next step. So it's like for transportation, that's it. And this is the easiest step. That one is done. So you're just converting pyruvic acid into acetyl coenzyme A is what this molecule is called now. Didn't make any ATP in this step, but you did take some energy away from the pyruvic acid molecules and give it to NAD to store, to use in the last step of cellular respiration. Okay. The third step, step 3 of cellular respiration, is called the Krebs cycle. Again, this step does require oxygen, just like step number 2. It occurs in the exact same place also. It occurs in the inner matrix of mitochondria. And then for the function, the modified remains of the glucose. So now glucose isn't really not glucose anymore. It's been broken apart. It's been split up. Lots of reactions happening to it. So now it is completely broken down. What's left of your glucose molecule is completely broken down. Now, after this step, there's nothing left. This step produces 2 ATP through substrate level phosphorylation. And during this step, large amounts of energy are collected and then stored for use in the last step. Okay. So this is a representation of the Krebs cycle. It is a cycle. So you start with an acid that is already in the mitochondria. You add the acetyl-CoA to it, acetyl coenzyme A. You go through the cycle and you end up with exactly what you started with. You end up with oxaloacetic acid. So it does go in a complete cycle. And the end product is exactly what you started with. So basically the coenzyme A molecule, remember I said it's just a carrier molecule. So it carries this acetyl group into the Krebs cycle and then it just breaks away and leaves. It is not involved in the reactions at all. It's just a carrier. So the acetyl group is going to combine with oxaloacetic acid and it's going to produce citric acid. And then from there, I mean, you just convert one acid to another through various reactions that are happening. Citric acid becomes isocitric acid, which becomes ketoglutaric acid. It just keeps going around until you end up with oxaloacetic acid, which is what you started with. During one round of the cycle, you can see that you're making a molecule of ATP and you're also reducing several coenzymes. So you're taking energy away from these acids and you're storing it with these coenzymes. And remember, just like with the first 2 steps, all of that stored energy is going to be saved to be used in the very last step of cellular respiration. Once you've gone through the Krebs cycle and both of the acetyl coenzyme A molecules have entered and been completely broken down, now there's nothing left of them. There's nothing left of the acetyl-CoA molecule, which was derived from the initial glucose molecule. It's completely broken down. So a quick summary of what's happened so far, and you guys can fill in the chart. We have the first 3 steps listed here of aerobic or cellular respiration. They are glycolysis, then step 2 is the oxidation of pyruvic acid, and then step 3 is the Krebs cycle, what we just talked about. What's happening, I filled that in for you. So you start off with glucose. It's broken down into 2 molecules of pyruvic acid in glycolysis. Both of the pyruvic acid molecules become acetyl coenzyme A in that second step. And then during the Krebs cycle, the acetyl group from acetyl coenzyme A is completely broken down. In terms of ATP, let's think about how many ATP molecules we made immediately from these 3 steps. During glycolysis, remember, you made 4, but you used up 2 initially. So your net gain of ATP is just 2 here. During the second step, you didn't make any. And during the Krebs cycle, you made 2. So far, we don't have a lot of ATP yet. In terms of the coenzymes that we have reduced, so just remember that when you reduced these coenzymes, you were giving them energy. So that means these are now really high energy molecules, and that energy can be used, and it will be used to make a lot of ATP. So for NADH plus H, we, during glycolysis, made 2 of them. During the second step, we made 2 also. And during the Krebs cycle, you made a whole lot. You made 6 total. For the FADH2, that was not involved in the first 2 steps. So you didn't make any during those first steps, but then during the Krebs cycle, you made 2. So all of those reduced coenzymes that you made during those first 3 steps are now going to enter the last step, which is called the electron transport chain. So basics about the electron transport chain: the step does require oxygen. It occurs on, let me go back and show you the mitochondria again. Do you guys see how there's an outer membrane that's shown in yellow? And there's an inner membrane that's shown in blue, and it has all these folds. It's folded in and out. The folds on that inner membrane are called crista. So for the location of the electron transport chain, it occurs on the crista within mitochondria. So it occurs on that inner folded membrane. Okay. So the electron transport chain is actually several proteins. They are shown here in this like brownish orange color. It's several proteins that serve as electron carriers, and they pass electrons down, and they're located on that inner folded membrane within the mitochondria. Okay, so the process of the electron transport chain is now you have all of the reduced coenzymes that came out of the first 3 steps of this process. They all enter the mitochondria if they weren't already in there. There are 10 total NADH plus H molecules that were reduced, and 2 FADH2 molecules. So these coenzymes are going to, they're going to now give away their hydrogen ions and their electrons. Remember, when they got reduced, they gained hydrogen ions and electrons, and that's what gave them energy. But now they're going to give up their hydrogen ions and their electrons. The electrons get passed down from one carrier to the next, to the next, to the next. And I'm going to explain this, but then I'm also going to show you guys an animation that helps explain it. So if you don't kind of get what I'm saying the first time, hopefully it'll make sense when you see the animation. And also, this is all written down for you in your outline, so you can go back and kind of reread it, and maybe it can make sense after looking at it a couple times. I know this is a little bit of a tricky process to understand. I get that. It often takes a couple of times going over it to really understand it. So electrons are getting passed along from like one protein to the next, to the next, to the next. And these proteins are using the energy that's kind of radiating off of these electrons to pump hydrogen ions out from this inner matrix out to this space out here. So they're pumping the hydrogen ions. And remember, they came off of those reduced coenzymes. The electrons and the hydrogen ions came from those reduced coenzymes, NADH and FADH. So just to explain where these things have come from, the electrons are passed along, and as they are passed along, they are giving off energy, which is used to pump the hydrogen ions out. Now you can see all of these hydrogen ions, you can see the direction that they're moving out, and now they all start accumulating out here in this space. So here is where you finally can see how ATP gets made. As hydrogen ions are allowed to diffuse back in, because once you have a high concentration out here, things always want to naturally move from where there's a high concentration to where there's a lower concentration. So their direction of movement is gonna be back in. They want to come back in once you've pumped them all out. They are allowed to travel back in through this structure, which is called ATP synthase. When the hydrogen ions move back through ATP synthase, it allows an ATP molecule to be made. Okay. So this kind of explains that last step. As hydrogen ions pass from this outer region back into this inner region, they go through ATP synthase. And as they come back in, it provides the energy to create a molecule of ATP. So here's the animation that I was talking about. These are your electron carriers. These are just proteins that are stuck in the membrane, that inner folded membrane. Here is the space all the way inside of the mitochondria, and here's the space that's kind of in between those two membranes. And these circles with the negative sign, electrons have a negative charge, so these represent electrons. So you can see as the electrons are passed from one carrier to the next, they provide the energy to pump the hydrogen ions out. Then, as the hydrogen ions are allowed to move back in through ATP synthase, ATP gets made. So look, now they come back in and you make ATP. So where do the electrons and the hydrogen ions come from? They come from these coenzymes, NADH and also FADH. So for every NADH molecule that you have, you can make 2.5 ATP molecules. And FAD, same thing. It just doesn't have quite as much energy. So you can only make 1.5 ATP molecules for every FADH2 molecule that you have. Okay, here's the significance of oxygen. There always has to be oxygen in the mitochondria. And after hydrogen ions have moved back in, allowing ATP to be made, the hydrogen ions and the electrons have been passed along. They are attached to oxygen. And when you bind hydrogen ions to oxygen, you can form water. Water is H2O. So if you put 2 hydrogen ions on an oxygen, you can just make water. And that water is a byproduct of this process because that is what happens. And here's why you have to do that, why you have to attach the hydrogen ions to oxygen to make water. If you have equilibrium, so if you have the same amount of hydrogen ions on the outside of the membrane as you do on the inside, you have equilibrium and hydrogen ions aren't going to be able to move back in. Did you guys see that happen? So if they're equal, there's no movement. The hydrogen ions are stuck out there. They can't move back in. And if they can't move back in, think about what's not happening. You're not making ATP. And you need these hydrogen ions to come back in so you can make the most ATP possible. So here's what you can do. You take these hydrogen ions away by attaching them to oxygen and making water. Now they're gone. Now you do have a difference in concentration. So hydrogen ions can move back in. Okay, so that's really significant. Why you have to have oxygen present so that the oxygen can bind to the hydrogen ions, carry them away, and more hydrogen ions can continue to move in through the ATP synthase. So you can continue to make ATP. So let's see, I just want to fill in this part of your outline and then I'm gonna stop there. So it explains that oxygen binds to the electrons, binds to the hydrogen ions to form water. The result of the electron transport chain is that you make 26 or 28 ATP from all of those hydrogen ions just moving in through ATP synthase. Remember, each NADH plus H molecule will lead to the production of 2.5 ATP molecules. So if you have 10, it's going to give you either 23 or 25 ATP. And if you read below, it explains why there's a difference. Remember, initially I told you guys there's a difference in the total amount of ATP that your cells can make based on what cell it is. So the difference in number of ATP produced is due to the need to actually use up some energy to carry the NADH that was made during glycolysis into the mitochondria, because glycolysis occurs outside of the mitochondria. However, if you're looking at cells that are in the heart, liver, and kidneys, they don't need to use any energy to transport that NADH in. You guys don't need to know that much detail as to why the number is different, but what you do need to know is where the number is different. It's in those 3 places, those 3 types of cells that you can make the higher number. So the heart, liver, and kidneys is where you would make in this process 28 ATP total from the electron transport chain, whereas every other cell in the body would make the lower number. Each FADH2 molecule will lead to the production of 1.5 ATP molecules. So if you have 2 FADH2, 1.5 plus 1.5 is 3. So that'll lead to the production of a total of 3 ATP. So if you're making 3 from the FADH2, And then from the NADHs, you're making either 23 or 25. You add 3 to that, so it's a total of 26. Or it can be a total of 29. And then you guys have to add in, so that's just the electron transport chain. But remember, ATP was made during glycolysis, you made 2. And then during the Krebs cycle, you also made 2. So add those in, and the total number of ATP produced from all of the steps added up is 30 or 32. And all you need to know is just where you make the higher number. Heart, liver, and kidneys is where you make the higher number of ATP. Every other cell would make the lower number. So this is total, once you're done with the whole process of cellular respiration, you get 30 or 32 ATP molecules. And at the bottom of the page, you can see the chemical equation for cellular aerobic respiration. That first chemical formula represents glucose. So you have glucose going in. It requires oxygen. You have to have some oxygen molecules. So you've got those. And then your end products are, I mean, the main thing is ATP, 30 or 32 ATP. Plus carbon dioxide and water. And remember that water is coming from when you add the hydrogen ions to the oxygen at the end of the electron transport chain, you make water. So those are your end products of cellular respiration. Okay, so that right there is the hardest part of Unit 4. It's all stuff that's happening like on the cellular level. So it's really kind of difficult to picture it. I know that's tough. So reread your notes, look at the PowerPoint. I think those animations definitely help with understanding the electron transport chain in particular. And basically, like for the 4 steps of cellular respiration, you mainly just need to know like what goes in, what comes out. So like for glycolysis, glucose is what you start with. That's what goes in. What's your end result? How many ATP are you making during each step? Like how many ATP are you getting right away? And how many of those coenzymes are you reducing during each of the first 3 steps? Those are significant because when you're reducing them, you're giving them the energy to store. And that's what's used to make all of this ATP during the electron transport chain, during this last step. So if you know that, your understanding of cellular respiration is good. Also know for glycolysis, it's the only step that can occur without oxygen. The other 3 steps, you have to have oxygen present or the process can't occur. All right, so I know that was a lot. That's kind of like an intense lecture. I will record another short lecture so that I can help you guys fill in parts of the other outline as well. And I'll have that up next week, probably early in the week. So you'll have it for over a week before you guys have your test on this, which is 2 weeks from now. So you have a while to kind of go through this stuff. Just a couple of reminders before I end.